seminar is part of our Digital Ag seminars, so for the semester. So, and then we are very, very fortunate today to have a very great speaker and also a friend. So we have today here Ryan Bartlett. He's a Chief Technology Officer for Pairwise. I know Ryan since many years in the past, previous life when we were working at Kansas and some projects on different companies. And then Ryan brings a wealth of knowledge going all the way from, and he's gonna talk about the presentation. I saw some of the slides, so I'm not gonna spoil anything. Or his path in industry from Monsanto, biotech, and then moving into management position in also nutrient fertility company, and now going into Pairwise. And he's gonna talk a little bit about his experience on gene editing and opportunities that we have here at Purdue to really partner with Pairwise. So with that, thank you, Ryan, for your time.
Thank you, Ignacio. I appreciate the invitation. Thank you all for taking a few minutes out over your lunchtime today to come hear about Pairwise. As Ignacio said, I'm going to start off just telling you all a little bit about myself. And I know that there's a mix of folks in the room, but I always found it interesting when I was going through undergraduate, graduate school, and postdoc, just the different career paths that people took. And I wanted to share a little bit about that with you to start off. And then I'll talk a little bit more about Pairwise. Some of you may be familiar with Pairwise, may be familiar with gene editing. I'll go into some details and give a little bit of background on a few specific case studies. So for me, I grew up on a farm in central Kansas, but like some farm kids, I think I couldn't wait to get off the farm and go do something different. So I actually went to University of Kansas, got a degree in genetics with a plan to go to med school, had been applying to med school, was ready to go into med school, and then started doing research and just fell in love with research. So I started my research career on a protein called calmodulin. So very, well, I guess across species, calcium regulation pathways. I was doing structure function work with calmodulin. In particular, that started in wheat. So it was kind of funny, farm kid in central Kansas that had grown wheat my whole life gets into basic research and starts working on a protein that we'd isolated from wheat. In particular, there's two methionines in calmodulin at the C terminus that under stress become oxidized. And it changes their ability to bind calcium. And then obviously there's a lot of downstream effects from that. So started my research in calcium signaling at KU. PhD from University of Georgia, focused on structure function, did NMR as part of my graduate work, but still all focused on calmodulin and then going downstream to some of the binding targets that calmodulin worked with. Continued that research at Vanderbilt Medical Center for a postdoc, where we were looking at the impacts of downstream regulation of calcium calmodulin with phosphokinases. And some of that had implications for Parkinson's disease. So was still kind of on the medical track when I was at Vandy, been postdocing for about 3 years and decided, it's time for me to really figure out what am I going to do. The academic path at the time was somewhat interesting, but I thought, you know what, I really kind of am feeling like I need to get back to agriculture. And so having done all this work in medical field and doing basic research for really human applications, I started looking around and thinking, how can I take my learnings and take it back to ag? There was, at the time, many of you won't know this because you're probably way too young, but there was a website called monster.com where companies used to post their jobs, right? And so I went on, randomly put my resume on there, and someone from Monsanto called and said, hey, we have a biochemistry position that's open in St. Louis. Would you like to come interview for that role? And I thought, okay, that's interesting. I can take all the biochemistry that I'd learned in mammalian systems and apply that to plants. So joined Monsanto in late 2005, early 2006. Worked this, what you see here is actually dicambo monooxygenase, the enzyme that's responsible for dicamba tolerant and now extend to soybeans. So I did kinetics on that. Also worked on drought guard corn when I first joined Monsanto, did bench biochemistry for a couple of years, led herbicide tolerance teams at Monsanto for a few years, and then went into commercial organization where I led specialty crops at Monsanto, including sugar beets, alfalfa, grain sorghum, winter canola, spring canola. It was a fun job because it was all of the things that nobody really cared about at Monsanto because it wasn't corn and it wasn't soybeans. So I learned a lot by doing that. Moved to, as Ignacio said, then moved to a company in Kansas City called Compass Minerals, where the organization had begun to build a specialty micronutrient portfolio, a large portfolio in Brazil, large portfolio in North America that has since been divested in South America to ICL, in North America to Coke Ag. But we developed planter box nutrient additives, seed treatments, fertilizer treatments, all focused on delivering micronutrients at the right growth stage for 10 different cropping systems. So that was a lot of fun. In my career at Monsanto, I spent a lot of time on germplasm traits and chemistry, 0 time on plant nutrition. So being able to round out the experience at Compass with plant nutrition was valuable for me. And then 3 years ago, moved to Pairwise. So I wanted to get back into something that was a little bit more cutting edge genetics play in crops. Pairwise, I'll tell you a little bit more in a minute, but came out of Monsanto. And I knew a lot of folks that were working there and had an opportunity to join as Chief Technology Officer and work with an amazing team of people to develop next generation traits through gene editing. So my passion really comes from that start of culture at the family farm. So we're in Russell County, Kansas. These are my great-great-grandparents on the farm in the dirty 30s. So I'm now 5th generation owner of my dad. And in Kansas, it's all about wheat, sorghum, alfalfa, a lot of those crops that I worked on at Monsanto, and now have the opportunity to work on again. So that's where the passion for agriculture comes from me, even though I thought I wanted to get away from it for a couple of years. So before I dive into the Pairwise piece, I just wanted to share a little bit more about my career path. I kind of gave you the color overview. But these are a few of the types of roles that I've had the opportunity to be in during my career and kind of what I've learned from each one of those. So for those of you that are thinking about what's next or, what's the path that makes sense for me as I move through undergraduate, grad school, postdoc, kind of what are some of the options? So as I said, started as a bench biochemist, really learned how to work in industry, which is a fairly steep learning curve. So going from, a lab setting or a postdoc setting even and going into industry, the metrics are different, right? And the timelines are different and the goals are different. And it takes a little bit of time to kind of change the way that your brain works. But I find it really rewarding because you're constantly thinking about the product. And so for me, it's all about how do we make products. Project leads, product managers, spent a lot of time in tech development, which at the time in Monsanto was taking the technologies developed in the labs and translating that to the field around the world. So a very humbling experience when you go from ag in the US, which is all about how can I make more dollars per acre, to sitting down with folks in sub-Saharan Africa or Central Africa or parts of Southeast Asia and talking about how do we produce more food for my family, to eat. It's a really great experience to be part of that ecosystem in ag and be able to have a small part of influence on that as we think about efficiency, nutrition, and all the angles that we can influence. At Compass, I had a few opportunities to be in the leadership team there, driving strategy for the organization. But then, like I said, at Pairwise, really getting back to technology and the opportunity to deploy that into the sectors that I love. So that's a little bit about my background. I'm going to go now and talk about Pairwise. So as I said, I think some of you are familiar with Pairwise, but a lot of you may not be. Pairwise was started out of Monsanto as a company that was focused on creating gene edits in row crops. Was founded with original CRISPR-Cas9 technology from MIT and Broad, really with a focus on how do we collaborate with folks like Bayer to create gene edits in their corn pipeline and their soy pipeline. At the time, it was also in canola and wheat. Do all the editing at Pairwise, but then hand those traits off to an organization like Bayer to put in their testing network. We're really focused also on foods that people consume directly. So I'll talk in a little bit about a gene-edited salad that we launched in 2003. That was the first gene-edited direct consume product launched in North America. We also work on things like blackberries and cherries. So we don't just do corn, we don't just do soybeans. We're really focused on the entire food network. And what we like to say when people ask, what is Pairwise? What do you do? We're really focused on making plants easier to grow and eat. Whether it's our food, whether it's food for agriculture, for hogs and for cattle, we're really focused on that. It's kind of our tagline. You're all very familiar with the drivers in agriculture. Don't probably need to go into a lot of this, but the areas where we believe gene editing can have an impact are listed here on the slide. Food security continues to be top of mind around the world, not just in the US, but I think there's a renewed interest even in the US as you look at initiatives from like Department of Defense and other departments where they're focused on how do we maintain security in our food system. Gene editing can help do that, not just by protecting yield, but also by mitigating waste. So as you think about indirect consumed crops like blackberries, there's a high percentage of waste from the time that blackberry is picked in the field, put in a clamshell until it ends up in your refrigerator. There's about a 30% attrition rate in that food supply chain. That's a ton of waste before it ever even gets to your refrigerator. So a big opportunity to improve shelf life. Human health. One of the things we're really passionate about at Pairwise is making foods healthier. And the salad example I'll give you in a minute, I think is a good illustration of that. But also just thinking about blackberries as an example too, very, very healthy snack that kids love to eat. They aren't very accessible year round. So we're working with companies to develop varieties of blackberries that can be grown in a variety of different climates so that we can make them more accessible, make them less expensive, and get a more nutritious option in the food aisle. Labor continuing to be a big pressure in a lot of parts of the world as it relates to labor shortages, especially in areas like California, where most of the blackberries we eat in the US are grown. And then just the supply chain. So as I already talked about at the beginning, it kind of comes full circle from food waste, not only about conserving yield, but also protecting our ability to maintain the integrity of the produce as it gets in the supply chain, but then opening that supply chain up to new geographies so we can make fruits and vegetables available to families that traditionally don't have them. We partner to make all of this happen. So Pairwise only owns genetics in one crop, and that's blackberries. All of the genetics that we work with come from other partners such as Bayer, Corteva. Sunworld is our cherry partner. I'll talk about that in a minute. But our model is bringing CRISPR tools to everyone to develop products that help to solve some of these global issues. And we do that through these partnerships, and I'll go into more detail. But as we think about just gene editing at its basis, a lot of people think that gene editing is going to replace breeding. We see the opposite. Gene editing and breeding are really made to go together. So we think about gene editing as just super fast advanced ways to breed for specific traits that we're looking for. We're using the potential that's already present in the plant to bring a trait forward that may have taken 400 years to produce through conventional breeding, right? So the watermelon, the seedless watermelon example, I think is a good one. Not that the focus was always on let's make a seedless watermelon for 400 years, but the outcomes of all of those breeding programs ultimately got us to the seedless watermelon that we enjoy today. We've been able to do this in blackberries in just a matter of about 5 years. So changing the time frame in which we can create products creates real value and opportunity for everyone. So we see gene editing coupled with breeding as the real opportunity. Create a trait that you can then put into a breeding population and forward breed with that to make it prolific throughout. So a big opportunity to partner with breeders. I'm going to talk for a minute about what Pairwise calls our Fulcrum platform. So this isn't just about the CRISPR tools. The partnerships that we develop with others outside of Pairwise are really focused on 3 major segments. The first one is trait biology. So if you don't know what phenotype you're looking for and what targets you want to edit to get to that phenotype, the tools are completely useless. So the big part of the equation for us is what do we want to make that creates value? And what's the specific target that we want to edit? And then what is that edit that we want to make? Is it a knockdown? Do we want to create a loss of function edit? Do we want to slightly tweak it through making specific changes to amino acids, or do we want to change the function completely? So that's the first, and I think the hardest part of all of this that we do is identifying the right target to start our editing platform. So we've worked in over 60 crops. We've transformed 14 crops and made edits in 14 different crops. I'll give you a few examples here in a minute. But then the tools that we use are proprietary to Pairwise. So it's something that we developed outside of the traditional Cas9 system. It's different than Cas9, different than Cas12, so we have full rights to that and we license that broadly. But then able to take these edits and deploy them at scale, that's where the partners come in. So we're not, we do what we do best, which is the two things on the left. We partner with people that can test and commercialize through their existing networks. That helps us to bring these things to scale as fast as possible. So that's the model that we use at Pairwise. So I alluded to this just a little bit, but how do we get from the start, the trait or the phenotype that we were looking for, all the way out to having to edit to characterize? This is obviously an oversimplification, but looking through a lot of crop science data to identify what's the trait again that's valuable. Some of those are things that we know are problems that we want to fix. The example you'll see here in the picture, that's a pitted cherry, right? Cherries are amazing, but why do you want to mess with the pit? We're working to remove the pit. So we know the phenotype that we're looking for here is no pit inside the cherry. Identify the gene that we want to start to target to look at edits for how do we remove the pit. AI is a big part of what we do here now. So we're using a lot of AI small data models to be able to look at very specific data sets to help us predict, here's the gene that you should be targeting. We then also use structural biology and AI modeling to try to predict what is the edit that we want to make and what's the resultant change in that protein. And sometimes that is a simple loss of function, but a lot of times, more often than not, it's actually tweaking the activity of that enzyme or that protein. Look at that sequence. And again, this is all very much AI driven. We have a phenomenal transformation team that has worked on these 14 crops, get them transformed everywhere from just traditional agro techniques to we do a lot of protoplasts now. So our teams do a lot of protoplast transformation work, introduce the tools through those different mechanisms, grow them out in controlled environment, and then ultimately hand them off to our partners to test in the field. So the tools that we have specifically, and I think everyone's fairly familiar with this, but if you look at the three main types of CRISPR tools that people talk about, the first bucket's nucleases and probably the most common. So if you look at the CRISPR Cas9 or Cas12a system, you go in and you just have a nuclease that makes a SNP, and you're basically removing a portion or creating, could be an early stop codon, could be a lot of different ways to just create a loss of function edit. So the example here is you're just removing a word, so the sentence kind of no longer makes sense. It's a traditional knockout. Base editing is, I think, where we have found the majority of the value come. So this is the ability to go in with a specific tool and make single amino acid changes within a sequence. So you can really go in and set up an allelic series within an enzyme to tune the function of that enzyme or that protein to get to the phenotype that you want. So you're going in and changing, in this case, there's a misspelled worst, and changing it to say worst, and you're making that very specific change through this base editor. Templated editing, or sometimes referred to as prime editing, is another way that we can go in and actually change the entire word. And so we can go in and change activities, we can change binding. This is usually looking at much larger sets of sequence where we can influence protein folding, not just a single amino acid change, but actually change the function and change sometimes the binding dynamics of downstream partners. So nucleases, base editing, and templated editing, those are the three types of tools that we're using at Pairwise. The same thing is true with Cas9 from MIT Broad, with Cas12 from the West Coast folks and what Corteva use. But all of the tools have a little bit different efficiency, and it's really key to make sure that you're pairing the right tool with the outcome that you're looking for, the phenotype that you're looking for. So core to this also, as I just said, is developing the right tool for the job. A lot of people will take a Cas9 off the shelf, utilize it in whatever crop that you're interested in, and you can probably get knockouts at a certain rate that may or may not be as efficient as you need. We have an in-house protein design engine where we're constantly iterating the designs of our nucleases, of our base editors, and of our templated editors. So we have a team that is just focused on taking what we call our shark nuclease, we call ours shark, and being able to tune the types of sequence that it'll bind to, being able to tune the efficiency of cuts for the intended application. So taking our shark protein and using it in corn, and then taking it and using it in soybeans, you're going to get a different level of efficiency, not only because your transformation system is different, but just the activity of that enzyme in a different species with different sequence, different binding parameters is going to change the efficiency. So we try to tune our tools for the specific species. And then those come out with proprietary base editors, proprietary diversifiers. Guide RNAs are huge in gene editing. So making sure that you have the right guide to identify the right sequence within the genome that you're targeting. We found that we get a lot of our specificity coming from AI-designed guide RNAs when we have the right data set to train AI on, and then gives us higher access to PAM sequences or access targets, and then pathways to bring those edits in and complete new editing systems. So this is a sub-team in the organization in of itself where their job is to constantly evolve the enzymes that we're using. So I'm going to move on from now talking about the enzymes specifically to talking about the outputs or the things that matter here. Within Pairwise to date, we've made about 11 million transformations. And as you think about transformation, you know, the number sounds big, but you think about throughput of corn, you're like, yeah, maybe that's not that big a deal. But I think for me, as I said earlier, that's across 14 different crops. That really isn't corn. Corn is actually a very small percentage of that. So we have a transformation group that is just focused on developing transformation systems in the crops that our partners want to produce traits in. When we think about a phenotype that we're looking for and getting that phenotype, we have a 72% hit rate. So let's say that we want to create a target for Asian soy rust in soybean. And this was one of the collaborative projects that we had with Bayer. We said, here's what we want to produce: a non-host resistant mechanism of giving ASR resistance in soybean. So as an example, across all of our portfolio, when someone comes to us with a phenotype idea, 72% of the time we're able to create that phenotype. Doesn't mean that it always becomes a product, because sometimes those phenotypes come with warts or pleiotropic effects where we have to figure out how to manage those or create the edit in a different way. But we're pretty proud about our hit rate. We've made about 81 potential products across those 14 species, and the IP portfolio is really important for us. And we have about 100 plus different patent families that cover the tools as well as the edits. Some of the key things that I'm going to talk about, we created last year, we put in the field the world's first seedless blackberry. I'll talk more about that. It's an interesting use case. Commercialized the first gene edited product in North America, the leafy greens that I'll tell you about. But we also partner with Bayer and Corteva. And I said this last night at dinner to Ignacio and Mitch. I think we might be one of the only facilities that has both Bayer germplasm, elite germplasm, and Corteva elite germplasm in the same building. So that's something we take very seriously. They take it very seriously, obviously, but we feel really humbled that they trust us to do their gene editing work. So as we think about identifying those products, a lot of times they come from our partners, but we're really focused in two buckets: growers and consumers. And I think all of us that work in ag have a similar mentality that a lot of times we're thinking about yield improvement, yield protection, right? That's the basis for a lot of the work that we do. So as we think about improved yield, whether we're looking at creating natural ways to just produce more with the same, that's one goal, or decreasing loss from things like disease, environmental stress, that's the other. But then on the consumer side, that's where we get into the nutritional piece, the seeds piece, and bringing those two things together in the middle is where we focus on things like food waste. So that's the lens that we look through for products at Pairwise. But most of the time, the collaborator that we're working with brings some of those ideas and we help to refine them. So I'm going to talk for a minute about the collaborations that we have at Pairwise. Started with Bayer. We now are in our second collaboration with Bayer. The first one, as I said earlier, is focused in corn, soybeans, cotton, canola, wheat, and really looking at yield efficiency and disease resistance across all of those different crops. So the team generated 27 novel traits that are now in different stages of testing in Bayer's pipeline, all field testing. Some of those are now going into their fourth year of broadacre yield testing in corn and soy, and I'll tell you about a couple of those examples in a minute, but really focused on, for the most part, yield and disease. The second collaboration that we have with Bayer down here at the bottom started in 2023 Most of you are probably familiar with Bayer's Preceon short corn system. So there's a breeding version of Preceon, there's a GMO version of Preceon. We're working to create a gene-edited version of Preceon that could be deployed more globally. So we're kind of in the middle of that collaboration. On caneberries, we have an exclusive arrangement with a company called Plant Sciences Genetics that is a breeder and germplasm provider for blackberries, raspberries, black raspberries, and strawberries based out in California. So I'll talk a little bit more about that. And then last year, we just signed an exclusive deal with Corteva to develop traits in corn specifically for them, all about, again, yield and disease. So, you think, oh my gosh, working with these guys on the same things you were working with these guys on, that's pretty difficult. But I will say they have both been amazing at partnering and understanding, what's off limits from Bayer and what's off limits from Corteva. But I think that there are so many good people at both of those organizations that they just want to solve the problems. And we're happy to be at the middle of some of that. So I'll tell you a little bit more about some of those. The other thing that I'll note down here, we do have a large, not yet publicly disclosed collaboration with a CPG company that's focused on editing in potato. And so this is something that we're really excited about and brings another direct consumer crop to the table. And we've had some great progress there. I'm not going to talk much about that today. A couple of other things that we are excited about is a collaboration with IITA to look at yam production. And so in Nigeria and a lot of parts of Africa, yam is a staple food. You can see in the picture here, they're long, sort of viney. You've got to trellis them. They're kind of hard to manage. They're really water intense crop. We're working with IITA to create what we're calling yam optimized architecture. So we've basically taken this vine and created more of a bush, made it more harvestable, made it more water efficient. And those are now going into the field with IITA. So very exciting stuff there. And then working with Summit, specifically on some sorghum in Nigeria and a few other maize and wheat targets that we have that our team's collaborating with them. With both of these collaborations, so this one is funded through Gates Ag 1, we give our tools to these organizations for them to use to create products without a lot of strings attached. So very important to us to work in crops that aren't just corn and soybeans. And then we also out-license our crops to other organizations that can do gene editing themselves. And that includes universities. I don't have any of the universities up here. We do have the Wisconsin Crop Improvement Center here. But for like Purdue, and I've talked to many of you over the course of the day today, we openly license our tools to universities to use for research purposes. But we also have a large collaboration that we announced a couple months ago now with Mars for gene editing specific in cacao. So cacao is a crop that has a lot of climate issues currently. Chocolate is really at risk. And so when we announced this, we thought, okay, are people going to freak out because now we're gene editing their chocolate? It was exactly the opposite. We got so much feedback about we're so glad that we can still have chocolate for many, many years to come because the cacao production system really is at risk of failure. I already talked about Summit and CGIAR. Tropic actually uses our tools and has been using our tools to create the non-browning banana that some of you may be familiar with. And then Solus and GenX traits take our tools to create traits for others as well. So we broadly out-license our toolbox. I'm going to get into a few specific examples here. Just to show the way that we think about transforming agriculture. I'll talk in detail about this corn example. I don't have a lot of time to go into the ASR example, but I'll tell you a little bit about it here because I don't have detailed slides in it. This was in collaboration with Bayer. Many of you are probably familiar that Asian soy rust poses a huge economic issue in South America in particular. So a lot of growers in Brazil that are growing soybeans are spraying 7 times with fungicide to try to control ASR. We were able to create a non-host resistance mechanism that's all focused on keeping chlorophyll active at a time when the disease is impacting the crop's ability to do that. So we're not actually influencing the infection itself. We're just having the plant through mechanisms of stay green continue to be productive throughout. These plants have now gone down from about 7 fungicide applications to 3. So it's not a cure. We're not fixing the problem, but we're helping it through unique ways. ASR is a really complex disease, and it's very hard to manage through a breeding population, so we're excited about that one. Blackberries I'll talk about a little bit more. Cherries I'm not going to talk about much today other than the learnings from blackberry that we'll take to cherry. But the cherry production system is also pretty intense. You guys probably all recognize, you see cherries on the grocery store shelf maybe 3 months out of the year. Good cherries, a lot of them are exported to China. So there's an opportunity for us to be able to make cherries that are available year round at a reasonable price. And then all of, this is really focused on removing pits. So I'm going to go into a little bit of detail here. The first one I'm going to start with though is this greens example that I talked about. So if you all go to the salad aisle in your local grocery store, there's usually 2 or 3 things available, and it's all usually iceberg or spinach or kale. Kale being one of the more nutritious ones, but a lot of people don't like the flavor of kale. Iceberg doesn't bring a lot of nutrition. Spinach kind of gets old. So company had done a bunch of consumer research back in like 2018, 2019 timeframe. And a lot of consumers said, we want salads that are more nutritious that our kids will eat. And so we looked at brassica diversity and looking at the opportunity to bring in different types of nutrition, but make sure that the flavor was there. So I'm going to talk in just a little bit of detail here about going into what ended up being our selection came from mustard greens. So if y'all are familiar with mustard greens, they tend to be used in the South a lot as a vegetable, but they're usually cooked. And they're cooked because they have a very pungent flavor to them. That mustard flavor is very pungent. A lot of people don't want to eat a salad that has that sort of pungent wasabi flavor to it. So we were able to go through select germplasm that had good nutrition, good color, good texture, good storability in the bag, and go through with gene editing and just fix the flavor. And I'm not going to go into the pathway detail here, but these have a pathway that produces this mustard oil. And we basically just went in with gene editing, and this is an example of a nuclease and just turned it off. So you could go from, and if you all have a chance to be in North Carolina and want to taste this in person, we have you taste the pungent one first and then taste the edited one, and it's night and day difference. So we launched this in 2023 through food service, meaning it went to restaurants, got great reviews from all of that, sold it in New York City through retail for about 6 months, and then have since licensed that to the Seminus portion of Bayer, and it's now being sold through their seed division. But this is just one example of making nutrition more accessible through gene editing, where all we had to do is turn off a pathway. No other pleiotropic effects came from this. We ended up making 13 different edits in the same plant, and we edited 9 different varieties to give you a mix of salads. So that was a pretty fun project. The next one I'm going to talk about in more detail is corn yield. And this was an example that came out of the first collaboration with Bayer, where the team was focused on just how do we create more yield. This wasn't about yield protection, this was about yield creation. And we looked at just kernel row number. And so if you look at inflorescence within the meristem or within the stem cells, this is all dictated by one gene called FEA2. And so we were able to go in and found alleles in the native germplasm that sort of dictates, here's how many kernel rows you're going to get. And this is true across almost every variety and genetic background of corn that you see. This is very, very well conserved. So when you have the wild type, you normally get 14 kernel rows. When you create a weak allele at this 477 location, you can get up to 18. When you kind of completely turn it off, you end up with this like softball looking thing that you can't put through a combine. So what we were able to do, and this is a specific example where we're going in and making very specific changes and creating an allelic series through base editing. So this is not just a nuclease. This is not just knocking out. We're actually going in and making an intentional allelic series at this P477 location in FEA2. And then what we get of that is the phenotypes that follow. So when you look at this, I think on the last slide, it was mislabeled. Wild type is 16. Our GUS control, so with no edit at the gene, is still 16. But then as we go through this allelic series and go from P to S to F to V, or completely delete it, we're able to really impact the number of kernel row numbers that are present on that cob. So this is what we like to call tuning, where we can go in with the editing tool and make a bunch of different changes, test them all in the greenhouse to find the one that's optimal. And actually the one that is in field trials right now is, these two are still in field trials right now, the 18 kernel row and the 20 kernel row. You get all the way up to here and you get some other malformation in the cob. And so with just going from 16 to 18 or 20, you've created about 10% more yield potential within that hybrid just by adding this as an edited trait. So this is now in 4th year of field trials with Bayer. The other thing that we wanted to look at though is we want to make sure that we haven't made the kernels smaller, right? Okay, great, you have more rows, but is it just smaller and so your hundredweight's gone down? So we really optimized for this P477S mutation where ear length stayed about the same as wild type, where kernel row count went up, but then also our kernel weight stayed up with moisture controls. So really fun project. Last year it was in Iowa and Minnesota. Now it'll be throughout the entire corn nest testing network across the Midwest. I'm going to switch over now from row crops and talk a little bit about blackberries. So some of you may be familiar with blackberries. It's a domesticated berry that tends to be grown in California for production in the US or in Mexico for export to the US. Blackberries are very long, brambly vines. You have to trellis them. They tend to be thorny. They're a very labor-intense crop to harvest. And that's why when you do see them in your grocery store, they tend to be kind of expensive. Probably most Driscoll's is the largest producer of blackberries. You probably see those clamshells most often, but really nutritious snacks. So very high in anthocyanins, a lot of great vitamin C. It's a really good snack that has great flavor. So we took the traditional blackberry plant with these long vines and we've edited to make what we call a compact version. I'll show you a picture here in a little bit. But these are the traits that we're focused on blackberry production. Taking that vine and creating a bush makes it easier to harvest, makes it easier to manage, makes it less water and nutrient intense because we've decreased the amount of biomass to produce the same amount of fruit. And ultimately, you can plant more plants per acre. So if you think about a blackberry bush or plant having these long vines that have to be trellised, you can only traditionally get about 1,800 plants per acre. What we've done is create this compact version where we're putting 5,000 plants per acre. So this is just taking the old corn yield story and replaying it in a specialty crop like blackberries. Making it seedless, pitless, that one's fairly obvious. And then we found a unique gene to edit out the thorns so that it's easier to harvest. So here's a little bit more detail about that. And here's the traditional blackberries. You can see they're very tall. There's a lot of biomass in the bottom third of the plant that doesn't actually produce fruit. And so through the edits that we've gone in and we've made these plants much shorter, we've doubled the amount that you're putting in a row, and we've reduced the row width by half. And so that's where we're getting up to this 5808 plants per acre. And these trials right now are going on in California and in Colombia. The fruit's actually being sold in Colombia. So this is now a gene edited berry that's on the grocery store shelves in Colombia. And we're seeing anywhere, depending on the season, about 1.5 to 2x yield with about a 30% reduction in water and nutrient. So you can just tell from the amount of biomass that you have here to produce the same amount of fruit, we're just being much more efficient. So it's a fun example for me because being a row crop kid from Kansas, I knew nothing about blackberries and it was a great learning experience. The seedless piece, I think, is also a fun story. So taking each one of these are called druplets on a blackberry. Those are all individual fruit. There's a seed inside each one of those. So that's why when you eat a blackberry, you get a whole bunch of those, seeds stuck in your teeth. Those seeds have a hard lignified endocarp around the seed itself. So what we've been able to do is go in and actually just turn off the gene that puts the lignin in the endocarp around the seed. And so what you see here is an unripe fruit, and we did this specifically because this is a stain for lignin. And you can see in the unedited version and the edited version, the berries look very similar. Can't really tell much difference. If you looked at a plant side by side, you can't tell the difference. The druplets look the same individually. But then when you stain for lignin in that endocarp, you can see on the edited version, there's no lignin there. When you actually eat this blackberry, you don't get any of that hard crunch from a seed, and it's actually juicier because you're letting a lot of the really valuable nutrients out of that seed, whereas opposed they're all traditionally they're protected by that endocarp. So that's another really exciting product that is coming forward. Currently in field trials in California. We're planting field trials in Colombia here in about 2 months. And so excited to be able to bring you all seedless blackberries sometime soon. And this one I forgot to mention is a combination of base editors and nucleases. So there's multiple targets that we're hitting here to create this. But so far it's done had really great performance in the field. The cool thing about this, though, is just thinking about what we call turning this into a franchise trade. So the seedless pitlessness across a lot of fruits has created sort of game-changing volumes in those segments. So you think about the seedless mandarin or the seedless grape, right? Those have all been incredibly successful products within their segment. Those were, for the most part, created through traditional breeding. So we're able to take some of the learnings from that breeding and now apply that in blackberry, but now take that learning from blackberry and apply that in other stone fruit. So that lignified endocarp and the genetic mechanisms to produce the lignin are the same in all stone fruit. So we're now going from blackberries and applying that to cherries in a partnership with Sunworld. So we now have some edits in our greenhouse in South Carolina where we've actually taken a cherry tree and we've turned it into more of a cherry bush, kind of the same story. We're just, hitting repeat. Makes it more easily harvestable, but it has also reduced the juvenility of a cherry. So traditionally, cherry trees need 3 years of juvenility before they will fruit. Ours fruit within the first 12 months. And we now have edits where we've been able to take the same genes that we had in blackberry and duplicate that in cherry to make a pitless cherry. The seed part we're still working with, but it's a pretty exciting project. But just because that mechanism is conserved, we can do the same thing in peaches, in nectarines, in mangoes. A lot of people have asked us about avocados. The mechanism is a little bit different in avocado, but when we talk to the avocado industry, what they want a pitless avocado, because the weight of what they're shipping, the majority of it's the pit. And so who knows what a pitless avocado looks like, if it's smaller or squishier or what, but just trying to take the learnings from one crop and apply it to multiple others. So for us at Pairwise, it's all about unlocking the potential that's already in the genetics of the crop. So we're not creating GMOs. We're going in and selecting specific traits that are already inherent to the plant that would come out eventually through traditional breeding and just doing it quicker. So we like to really focus, as I said, across geographies, across crop types, and we do this through partnerships. We're only successful when our partners are successful. And that's everyone from the Bayers and the Cortevas of the world to you all here at Purdue. So we're looking for partnerships in all parts of the ag spectrum and want people to have open access tools so that they can make the products of the future. But back to, we just want to make food easier to grow and eat. We're focused on nutrition, we're focused on availability, and we would love to get your thoughts and develop collaborations and partnerships with all of you. So with that, I will stop and take any questions. One of the things that I wanted to note here for the students in the room or for faculty that may have students, please take a look at our website. We do internships every year. Last year we had 15 interns, and it's usually a mix of undergraduates and grad students. The internships are based in either Durham, North Carolina, or Raleigh, North Carolina, and Research Triangle Park. And we have everything from computational biology interns to folks that intern in the greenhouse, or even last year we had an intern in the field in California. So we usually post these sometime around March or April, and I'll send Ignacio a note when we do post these. But if any of you all are looking for internships, we'd love to get some interns from Purdue. We have a lot of NC State interns. We have a few Duke interns, but we don't really like them. But no, it's great. We've had a couple folks from K-State, so we'd love to get some Purdue folks with us too. But with that, I'll stop and see if there are any questions for me.
Well, thank you. I enjoyed your talk more than about any other talk in recent months.
Well,
And
thank
you're
you.
an excellent communicator. So my question is, you say these are not GMOs, and I'm not a plant breeder, and so, but they are artificial genetic modifications, right?
Sure.
And so explain that and how you get around that label and such.
Yeah, and it's an evolving regulatory framework, but in the I'll give you the example for the US. So to be considered a GMO, you're putting in some exogenous DNA that's not inherent to the genome. So traditional genomes contain a transgene, expression elements, not only the gene that you're overexpressing. With gene editing, we're manipulating the DNA that's already present in the plant.
Mhm.
So I think that you could, argue that there's a gray area of whether or not that is synthetic.
Right.
But from a regulatory perspective, many regulatory bodies around the world have basically said that's just advanced breeding. You could get the same outcome over time through breeding by just doing cross after cross after cross after cross. A lot of, breeding programs tend to throw in some form of mutagen, whether it be UV or something else, to create these types of alleles.
Mhm.
And so we're just doing it faster.
Okay.
Yeah, but I think it is critical. We talked a little bit about it this morning with the group that, these have to be distinct from GMOs, and most regulatory bodies agree. Europe is still in process of their determinations. Mexico is still in process. Mexico, has a moratorium on growing GMO corn. But with their new administration, they've come across and basically started to agree that they don't see gene editing to be the same as GMO, but every country has a little bit different perspective. So.
So I noticed that a lot of the crops and the plants that your company focuses on are ones that are traditionally not very easy to transform, or if they are, I know it's very accession specific. And as you know, to be able to make your edits, you have to have some way to deliver it. Like I said, those delivery systems are known to be very accession dependent. How do you guys get around that? Like,
Sure.
do you have something in your pipeline that you do it, or do you work with?
Both.
Okay.
I will say on the woody species in particular. So woody species tend to be very recalcitrant to transformation. So we've developed an in-house proprietary method that's based on roots, where we have been able to take the same transformation method for woody species, transform cherries, transform peaches, nectarines, blackberries, raspberries. So this same method has been working for woody species. For non-woody species, we're probably not as good at that as maybe folks here, even after hearing from some of the things the teams are working on this morning. But we try to optimize the transformation system because it's a balance of transformation efficiency and editor tool efficiency. So if your transformation system's really, really good, your tools can be kind of crappy and you'll still eventually play the numbers game and get an edit. But if your edit efficiency is really high and transformation system's not so great, you can still get that edit. We try to optimize both so we can have as much throughput as possible. But yeah, it's a good question because that's the hard, not only finding the target and identifying the edit you want to make, being able to transform the plant is so critical. Absolutely.
Thank you.
Thanks so much for this talk. You mentioned a couple of times that some of the exciting elements of this are being able to breed for different kinds of plants, different kinds of farm ecologies. In the specific examples that you talked about just now, it seemed like a lot of these were still fairly monoculture models, fairly capital intensive farms, which might not always apply to some of those stakeholders you were mentioning in these other areas, right? So I was curious what you see as the role of something like gene editing and the work you're doing for these different kinds of farm ecologies and how that would fit into a different sort of model of farming for some of those other stakeholders.
Yeah, it's an interesting question and something that we've recently been contemplating with some of our partners in other parts of the world. So when you think about, and you're absolutely right, we're focused on production crops right now for the most part, but looking at using these tools to make cropping systems begin to blend together, I think there's a lot of opportunity there. Where we kind of fall short in that area as a startup company is we don't have a lot of funding to go out and do some of that on our own. So
Sure.
We're constantly looking for partners. Gates Ag One has been a good example of this exact question where they're asking us, what can we do to make some of these other non-traditional ag ecosystems better? And so whether we're looking at mixed crop production, monoculture type production, and just making things more compatible, there's some edits that we've started in our pipeline for some of those crops that tend to be grown together to just try to make them more compatible. But yeah, it's the right question. Long term, I think it's harder for us to figure out how to solve it because we don't have like a Bayer or a Corteva sitting there with a checkbook saying, here, we'll give you this much money to go do this, right? We're going to have to rely on other organizations.
Really enjoyed your talk. If you set aside the transformation difficulties species to species, what sort of tweaks have you made to make the editing efficiency be
Sure.
more efficient
Yeah.
species to
Yeah.
species?
And there is some variability. So with nucleases, it tends to be more consistent. But if we look at our shark nucleases in corn, we push upwards of 80% efficiency getting edits in our system. We take that same nuclease and put it into blackberry, and that efficiency went down to about 30%. So what we've found is really going in and engineering PAM sequences and engineering other components of the binding aspects of the nucleus itself, and we can bring that efficiency back up to 80%. So it's not always perfectly translatable from species to species, but we have found that we're able to go in and re-engineer slightly portions of those nucleuses to make them more productive in other species. But it gets back, Steve, to that balance of transformation efficiency and tool efficiency. And sometimes good enough on tool efficiency is okay, because we will just play the numbers game and try to get throughput.
Thank you.
So in terms of non-GM, are you doing this through protoplasts? And if you are, what is the subclonal variation? Or are you doing this through breeding out transgenes? Or are you doing this through transgene-free editing initially?
Depending on the crop, it's all three. And so we have protoplast systems that we're using in blackberry and potato. Those give us DNA-free, what people call DNA-free methods of
And subclonal
transformation.
variation with that?
Yeah, we continue to improve that, and this is in a collaboration with NC State, actually. We've improved the variation that we see to the point where we can start to get products coming out of the protoplast system. I would say has been more difficult than what we thought it would be and has just required a lot of iterations. We have also done crossing out of some of the transgene work when we make traditional transgene type of edits. But then the root method that I alluded to earlier is true sort of DNA-free type transformation. So we do all three methods.
Well, I was wondering, like you mentioned, that about the edits that result in the decrease of the pungency of certain plant crops. I was wondering, does it have a negative effect on like pest control? Because I imagine that like insects don't like the pungent taste, of course.
We had that exact concern. So some of the early field trials that we did were just looking at plant pest issues. And we didn't see any uptick in pest pressure. There were a couple of cases where, so we would grow these greens next to all other standard greens. There were a couple of cases where actually the other greens were attracting insects and then they would move into the mustard, these non-pungent mustard greed production, but not as a result of the lack of pungency. So we were concerned about that, but it didn't actually turn out to be a problem. Thanks.
Thank you very much for a nice talk. So I believe at Pairwise, using agrobacterium strains is a core part of a pipeline, right?
Sorry, I couldn't hear you.
At Pairwise, using agrobacterium transformation is a core part of a pipeline. And then you guys seem to transform a lot of plants. So I'm kind of wondering, based on transformation efficiency, how do you determine the right strains to use with certain crops? Because you're doing a lot of transformations.
Yeah, that's a good question. I probably don't have the great answer for that because I'm not the transformation expert. But it comes down to specifically what's the crop that we're working in. And then a lot of that, maybe we find from our collaborators where they've had good success, but a lot of it for us is trial and error when it comes to using agro. That's why we're more focused on some of these other systems now, like protoplast and this root-based method where we're not using agro. Chris.
Thank you for your talk. Is Pairwise or any of your partners working in more like, I guess, breeding tools area? So I'm thinking like double haploid induction or sterility systems, or is that an area that you're interested in expanding into?
It is, you know, sterility is something that I think the industry continues to be focused on for a few different reasons. So we have some, I'll call early stage projects going on in sterility. No real data or outcomes there yet, but you know, also just looking at other ways to overcome, you know, looking at apomixis, looking at other things that we can influence through editing. So I think that's, for us, that's kind of the next exciting wave of things that are coming from editing, is ways that we can just influence breeding in general and make it more efficient.
Thank you.
In tropical countries like Nigeria, you mentioned, I know that tropical condition favors microbial growth and like metabolic activity. How would gene editing be made efficient for that purpose?
Yeah, I don't know. There's a lot of, so Tropic is one company that's focused on improving tropical crops from a pest disease and pest resistance perspective. We don't do a lot of work in that directly, but I think there's multiple ways that you could influence that pest's you know, ability to hinder production of those crops. So as I said in soybeans, we looked at non-host resistance mechanisms, which won't always apply to probably to the types of things that you're talking about. But I think when you look at the editing toolbox, there are so many different parts in that pest you know, interaction, where you could go in and change something to maybe make it, if we're talking about a virus, less virulent, or have different pathways that don't necessarily bind, or improving host immunity, turning on our genes perhaps that maybe have become you know, ineffective. So I think there's a lot of different ways to do it. I don't have a really good specific answer for your question though.
Thank you.
Just one more question.
Okay.
So you mentioned about like internships and stuff. What about PhDs like me who are looking to defend next semester and is going to be on the job market? Do you anticipate Pairwise having any openings, or do you have any advice for students looking to get into the next step?
So Pairwise kind of has openings on a rolling basis, just depending on need. You know, I think making connections with companies, regardless of, if you know what your dream job is, find someone that you know at that organization and find a way to get introduced. But just developing your network, I think, is really key. Because if you, let's say you did an internship at Pairwise and get to know that team really well, and then you're looking for a job after that, but maybe there's not an opening at Pairwise, our collective organization has, you know, a network that spans geographies, spans companies. So I think getting into that is a really helpful way to help you just grow your network and find positions, because then we can speak on your behalf and say, we know, you know, she was a great intern, she's gonna be a great performer, we would recommend that you hire her for this type of a role. So I think internships are a really valuable way to grow your network. Yeah.
Thank you for your wonderful presentation. I also, my question is related to internship as well. So I'm working on like a nano delivery system in plant. So I also curious about your internship position about the transformation skills, but you talk about that you are collaborating with other companies. So I wonder you have some position about the transformation roles.
We do. Last year, I think we had 3 interns in our transformation team. And the transformations that, for all of the partnerships that we have, we do the transformation work in-house. We may partner with them on the development of the system, but all of that is done in Research Triangle Park at Pairwise. So yeah, we're always looking for interns in transformation, because that is kind of the linchpin to making all this work.
Thank you.
So keep an eye out. Like I said, I'll share with Ignacio once our internships post. But we would love to get some of you to come down to North Carolina for a summer.
Thank you so much.
Ryan, thanks for the presentation today. Appreciate that. I'm an agricultural economist, so several things you mentioned, I have no idea what we're talking about, but I know in general what we're doing is pretty cool. I'm interested in as a startup company, to the extent you can share, what's sort of the business model?
Sure.
How do you get investors interested in investing in this?
Yeah, it's a good question. It's something as a leadership team we contemplate almost every day. Being almost 8 years old as a startup means that we have to start looking for exits probably. So most of the money that we have taken has been venture capital money, and we're lucky enough to have institutional investors as well, like Bayer and Corteva are both on our ownership cap table. And so I would say if we rewind pre-COVID, going out and asking for money to do this sort of stuff actually was kind of easy.
Yep.
And so gene editing was, big hype. Everybody's like, we want to figure out how to invest in gene editing. So I would say for the company, the Series A, the seed was predominantly done by Monsanto. Series A was predominantly leaps by Bayer, and those institutional investors. And we had Temasek come in, we had Deerfield, who is not a traditional ag investor, but then Element. They've all been great every series to bring in fresh capital. I think what we're finding now is really hard is just how do you take it to the next step? And, everybody, the easy button is, oh, well, Bayer or Corteva will buy you someday. I think that's less and less likely as things play out, but who knows? IPOs are really hard these days too, because not many IPOs are very successful. If you back up a few years and you had companies like us going out and getting $1 billion plus valuations on an IPO and then not being able to deliver on that. So I think for us, it's more about telling the product story and less of the technology story. Even the technology makes it happen. And these collaborative partnerships are huge because we can show that we focus on the piece that we do best and we let someone else do what they do best. And that model, I think, is really amenable to bringing together genetics and technology. It's not dissimilar to what happened in the mid-90s with, you just look at what Monsanto did with the Roundup Ready trait. They couldn't get corn companies to take it on. So they just started buying germplasm companies and then did it that way. So I think we're kind of on the verge of how gene editing gets included outside of the major row crops by combining germplasm and the technology. But it's tough. It's a tough spot to be in. All right, thank you all very much. Oh, one more.
One quick question. Is there any possibility of blackberries bearing in the first year, or is it always going to be second year?
Yeah, I didn't get into that, but
That's all I came for.
These blackberries are primocane.
Sounds personal.
So we've also done the same thing in black raspberries and created primocane bearing black raspberries. Yes.
Where can I get them?
Well, right now you can't.
But, okay, carry on. Sorry, thank you.
But yeah, that was one of the keys to making this complex, is we've also influenced flower timing. So our blackberry crop is also about 15 days earlier than a traditional floricane crop from the same variety. Great, thank you.
That sounds like it's all.